Hello and welcome to a lecture on social cognition. I'm Vero Jääskeläinen, professor of systems neuroscience in Aalto University, Finland. And in this lecture, I will tell about social cognition and the neural basis of this very interesting function. And let us go forward. To get the best out of this lecture, I would suggest that you would go and download my book from bookbong.com and read in advance. Stop now and get this book, read in advance, chapter 12, and you will get already some basics, some basic concepts, and then this lecture can build on those concepts. There will be some repetition, so you can also follow, I'm sure, without this advanced reading, but it's just gonna facilitate learning a great deal. And this is also otherwise a very useful book. It's written to be very easy to understand, and it introduces the basic concepts in this rather large research area of cognitive neuroscience. Something other that will support your learning is to use learning diaries. After each lecture, there will be, now there's already one, and there's the second, and there's going to be third, and maybe some more. Let's see how this goes, that I will post on YouTube. So after each lecture, it is beneficial for learning to spend a few minutes listing keywords that come to mind on the question, what did I learn during this lesson? What were the open questions that you found interesting? Alternatively, you can write a short summary of what you learned, what was interesting, this alternative to keywords. Whichever way suits your purpose is best, but this facilitates learning, and that you pause and you remember back, okay, so this was interesting, that I learned, and then you get the repetition via writing this learning diary. Then, of course, as you head for a good night's sleep, after such a learning experience, we know that then during sleep, these memories consolidate. Irrelevant materials are cleaned off, memories are crystallized. So also remember, study hard and then sleep, rest well. So there's a good outcome. Okay, but so what is social cognition? It deals with, there are many different ways to define this concept, but I'm taking this approach. So how persons process information about and predict actions of others in social situations. So this is about understanding and predicting others' intentions, emotion, and goal-directed behaviors. And of course, then it also has to do with adjusting one's own behaviors so that one acts in a smooth fashion in the environment. And so also social cognition is very much goal-directed perception, and that you will be paying attention to the social interactions that are somehow relevant to what you hold in value, the kind of tasks you're engaged in at any given moment. And so just to illustrate how beautifully our brain works, I took this image from the internet, and for most of us, it's very easy to see what's going on in this picture. What are these two persons here? Where is their attention? What is the role of this person here? What's happening? And you just need a second or two to figure all this out. But if you stop to think about this for a moment, This actually requires very sophisticated information processing to figure out the relationship and where their attention is and what's going on. So this seemingly trivial task is not trivial at all. It's very challenging for the brain, but the brain is set up to do this kind of tasks, to read social situations. And in fact, as advanced by my colleague Robin Dunbar and his co-workers in Oxford, so development of neocortex, and I would say especially prefrontal cortex, prefrontal cortex is very important for social cognition. This development is predicted by how many social relationships a given species has. So here with the squares, you can see monkeys, and you can see neocortex ratio. The more neocortex, the larger the mean group size of these different species of monkeys and these different species of apes. So there's a very strong correlation. The larger the group size, the higher the neocortex ratio. And so maybe it was previously thought that general intelligence is something that goes hand in hand with the development of brain, especially development of more cortex. The more cortex one has, the more intelligent the species. But there's even a stronger predictor. How many meaningful social relationships do we have as a species on average? The larger our neocortex. So this is very interesting, and this led to this social brain hypothesis, that perhaps the biggest tasks that the brain has been set up for is figuring out social relationships and how to navigate the social environment and the world. Then something which I think is a very important landmark finding by Jacoboni and colleagues, and here's a picture from their publication in Neuroimage in 2004. There is this so-called resting state or default mode network in the brain, and why is it called the resting state network? It's because it shows higher activity during rest than simple tasks. And early on, this amazed scientists. Okay, so the subject is in the fMRI scanner and is resting between these simple tasks. And then they looked at functional connectivity and activity in the brain, and they found that there are these specific structures that really operate during this rest more actively than during the performance of these simple tasks. And it was called the resting state network. And then it's something that has been a topic of investigation. Why is this so? And especially you can see in default mode network these dorsomedial prefrontal cortex and precuneus regions, also inferior frontal gyrus has been thought of, and DPJ, the temporoparietal junction, have been associated with default mode processing. But so here, in this particular study, subjects were watching movie clips showing social interactions. And then, in contrast to the simple tasks, like arithmetic tasks and so on, or simple attentional tasks, visual attention tasks, and so on, there was higher default mode network or resting state activity during the watching of social interactions than during rest. And this raises the question, is the resting state network or the default mode network, in other words, activated due to social daydreaming in the situation that the experimental subject is there, the scanner can rest between these simple tasks. And then this points out that, okay, maybe the mystery has been solved. This very fascinating network of brain structures, maybe it has to do with social cognition. Okay, something that sheds more light on this, what is this default mode network doing, is theoretical work by a colleague of mine, Moshe Bar, and you can look up details in this very interesting publication, The Proactive Brain Memory for Predictions. The idea here is that as we navigate the world, we form quick associations about what we're encountering. And then these associations build the schema that allow us to navigate the world. And here it seems that the interplay between the medial structures of the default mode network and hippocampus or medial temporal lobe here, there's an interplay during this associative process. And then it's further postulated in this theory, I think quite convincingly, that this same network actually during rest, it goes through different kinds of scenarios and during daydreaming and builds these mini scripts and stores them. So then when we encounter in social life something new we haven't encountered before, but we have, thanks to our daydreaming, some ready-made scripts. Okay, so this situation occurred, I will behave in this particular way. And this, so our brain is all the time preparing. When we're not engaged in some other activity, our brain is preparing ourselves a lot of that time for future social interactions. Also, something which is really crucial for social cognition are eye movements. And it turns out why eye movements, we're gathering information from our surroundings all the time in a goal-directed manner. Of course, it's guided by what we need to pay attention to. And here you can see a picture which was used in a very I would say pioneering study in 1967 by Yarbus. And the experimental subjects were watching pictures like this, pictures of faces, and then the experimenter was recording eye movements. So the eye movements take place in these brief stops, and then there's quick transitions between these brief stops. And actually, as you know from elsewhere, probably our visual processing is such that we have quite a tiny area that we can process in our visual field accurately at any given time, and we need to move around our gaze to build the complete picture, or actually, refine the mental picture we have in our mind of what there is out there. And so what these researchers found was fundamentally important. These fixations predominantly take place at the locations of the eyes of the pig and the mouth region, or the eyes and the mouth region. They gather most of these fixations. And this is where we have this information about social signals of others. What kind of intentions do they have? Where do they look? Gaze direction is already something that tells a lot about the intentions of another person. Also, emotionality, these muscles in the face, smiling, frowning, and so on. This tells subtle signals about the emotionality of the person, and this is what we normally pay attention to. Then I put here a nice YouTube link, and let me do it this way. I will not tell you what this link is all about, but I recommend watching. It's a very interesting link about eye movements and social cognition. Then there are some findings that persons with autistic personality features, high loadings in autistic personality features, and neurotypical persons differ in where do they look at. For example, in a photograph or video, and here's just an example from an early study. This is from research by Baron Cohen. And the viewer with autism, as you can see, is looking at the clothing. This red curve represents their eye movement patterns, the clothing of these protagonists in this photograph, whereas then the neurotypical persons, they really fixate on the face, the eye region especially, but also somewhat to hands especially of these two persons, but also the third person on the background. So the neurotypical brain is quickly figuring out, what's going on in this picture, what kind of a social situation, what is the relationship between these different persons. We come up with hypotheses. But the autistic persons, they somehow are not interested in this, but rather look at, for example, the clothes or some background in the image. And I have this one psychiatrist colleague some years ago. He gave a presentation where he said that sometimes he gets these patients, autistic patients, and then they very intently look at his face and eyes, make a very long eye contact, and then he asks them, can they somehow get some information by looking at him, his eyes, and so. And these patients then reply that how could this be possible? Of course, he's not getting any information about the intentions or emotions of the psychiatrist from by looking at this face, eyes, and so. But he has been taught that this is polite, to look at the eyes of the other person. Okay, so then a study that we did some years ago on social perception, and this is now giving you some idea. I mean, presenting these individual studies, and throughout this lecture and these lectures, and the idea is that you will get an idea of how these experiments are built, and then also that you will get an idea of the strengths and weaknesses, and how to do better. Many of you who are going to listen to this lecture will face this situation of running your own experiments, maybe for master's thesis, maybe for a doctoral dissertation, and beyond. And in this particular experiment, we had these different categories of social actions that were included social features like faces, bodies, biological emotion, goal-oriented action, emotion, social interaction, pain, speech, houses and places and objects. So houses, places, objects, rich emotion, non-goal-oriented action, no social interaction, non-human sound. They were these non-social feature control videos. So we had these social feature videos and non-social control videos. And these are short videos, just maybe 20 seconds. And okay, so I'm not going to play this because of potential copyright issues. But here in this clip, there are these two persons discussing. It's such a short clip that you can't really figure out too well what's going on. But anyways, we'll have emotions and, exchange of words and so on. And then as an example of a control video, there's this airplane flying over a field and, spraying some kind of substance on the field. I remember there was, when I was giving this lecture in Aalto University, there was one student who said that they actually feels angry at seeing this kind of spraying of toxins, and this, of course, underscores the importance of asking the individual subjects afterwards. How did they feel? How did they experience these different kinds of videos? So it's always when you're running neuroimaging experiments with naturalistic stimulus, the videos, it's very important that you not only have the information about what's in there in the video and what is the brain activity, but also the subjective interpretations, experiences of the experimental subjects. And so we ran this experiment, and then there were two different raters saying when there were faces in these videos, bodies, biological motion, and so on and so forth. And you can see some were very easy to rate by these volunteers. So the inter-rater reliability correlation was 0.94, so almost total agreement of when there is pain in these videos. But then for something else, like goal-oriented action, there was less agreement. The correlation was 0.6, which means that 36% of the variance between these two ratings was explained, one to the other. And this is an example, is yet another example of the importance of paying attention to, what will they rate afterwards, and what is the instruction to rate. So I mean, the more vague the experience that you want your experimental subjects to rate, the more careful you have to be with the instruction. So if you want them to rate, did I feel this other person's pain? It's very simple, it's straightforward. There's going to be very high reliability, validity. But if you rate something else, like did I anticipate something is going to happen, very soon, then this rating will drop down. And so here you can see just the control analysis and correlations between feature ratings. And here you can see, there were some correlations that were quite high. So whenever there was social interaction, there tended to be speech, 0.54, but still, there's sufficient independency on these ratings. And so then there's this red text that today there's increasingly possibilities to use automated machine vision annotation tools, and then this is the topic in one of the seminars. So so this is actually a relic of a previous teaching session, this referral to seminars, but the statement is really true. Thanks to the development of autonomous driving vehicles, we have these machine learning algorithms that can really pick up from these long video clips, different things, very automatic fashion, like traffic signs, persons, and these algorithms, they can be adjusted and trained for various types of visual features. So with them, we can actually get a fairly good idea of what there is in each movie clip at each point of time. And then, of course, we can use this information compared to the brain activity. And then if we have these subjective experiences from the subjects, and we might also record eye movements at the same time as they're in the scanner watching movie clips, so we can get a fairly interesting idea of what they're paying attention to, what there is in the movie, what's going on, and so on. So this is how it works. But here we ran rather simple analysis. So we basically just looked at across different regions of the brain, so how many number of categories were these different brain regions responding to, and especially a number of social categories in this case. And you can see that there's the superior temporal sulcus, and also TBJA, and I would say there's also area MT involved, and this posterior temporal occipital junction areas on the lateral aspects, as indicated by the red color, they responded to all the social categories in these clips, 8 clips. Yes. So and for example, the STS, the superior temporal sulcus, the posterior aspect of it, failed to respond to the non-social categories. So then we even entitled the paper that this is the hub for social perception. And also, in addition to just looking at the activations, we ran something called complex network analysis. And here, I think this is the first ever, as far as I know, from the first ever experiment with complex network analysis of brain activity. It comes from a lab in Mallorca, Spain, and it's a fairly simple idea. You correlate I mean, for each brain location, you get a time course. Then you calculate correlations between these time courses, get a correlation matrix. So brain region against one of them, you can see here is the correlation is 1 on this, because each region, of course, the time course correlates perfectly with itself. And then you just threshold this matrix, and then you build this network. You extract the network, and you can get information such as what is the strength of this connection between 2 different. I mean, the strength of correlation. I mean, of course, this is only correlation information that you can never get from functional neuroimaging data. There are some algorithms that sort of estimate to what extent the history of brain activity in one region predicts the present state in another brain region, and it's called the Granger causality. But even that is correlation based, so this is something important to keep in mind. But nonetheless, we think that this gives a fairly good approximation, fairly good estimate of what these networks are. And so you can see that some areas are very nicely connected with one another, and some areas more weakly. And then you also see some regions that are connected very strongly with multiple other regions. So these are hubs. And then you can build your complex network. And so then we did this in our study. And here you can see an estimated functional complex network during social perception. And you can see the connection strength from 0.43 to 0.78. There's the node degrees. I mean, how many other nodes is the node significantly connected with? You can see there's, for example, this region here, which is only connected with one another region, but then there are these hubs that are connected with many other regions. And for example, this this STS in the right and the left. The posterior STS in the right and the left, they are connected with just multiple different other regions. The solid circles here represent right hemisphere, the striped left hemisphere, and then we could identify these different subnetworks: frontoinsular, frontotemporal, frontoparietal, and temporoamygdalar subnetworks. And then we looked at where which subnetworks activity time courses actually correlate with which social features, and we see frontoinsular correlating with emotions and pain, frontotemporal with pain, emotions, social interactions, and frontoparietal with bodies, biological movements, goal-oriented movements, and pain. And then this temporal amygdalar network correlated with cases, social interactions, and speech. And why is it important to understand brain connectivity? So traditional neuroimaging studies focused on the mapping of involvement of brain regions in various tasks. And any given brain region is, however, involved in multiple different functions. So this is a very important principle to keep in mind. And rather, the input-output connections of a particular brain region may give rise to the functionality, very much. I mean, there's some more recent theoretical work from a colleague of mine, Amir Mehdi. You can look up his work. It was in Trends in Cognitive Sciences a few years ago, where he presents some very compelling evidence that actually, yes, these areas are functionally specialized to do certain things, like inherently functionally specialized, not only because of this connectivity, but connectivity also plays a key role. So it's the combination of these two factors actually that seems to be the key, and so it is really important to understand connectivity of the brain. So, but something we saw, these short movie clips perhaps did not require theory of mind or mentalization, because we saw lack of activation in ventromedial prefrontal cortex, and this is sort of the hallmark region where we see a convergence of seeing activity whenever there's a theory of mind task. This is from an adaptation from a meta-analysis by Carrington and Bailey in 2009, where they looked across a wealth of different theory of mind tasks, and they saw that there was involvement of the orbitofrontal cortex, medial prefrontal cortex in 92% of the studies, and this is drastically lower amount of studies in other cortical locations. The temporoparietal junction, superior temporal sulcus, these regions that had to do with the sort of what we call the social perception hub. It's only involved about half of cases in about half of studies that looked into the neural basis of theory of mind across different theory of mind tasks. So really the orbitofrontal cortex, the ventromedial prefrontal cortex, seems to be the hub for theory of mind. And here you can see, yes, this posterior STS is involved in theory of mind, but okay, so there's something that these short video clips failed to do. They failed to drive these very core theory of mind regions of the brain, and then maybe these short clips didn't so much call for mentalizing. And okay, so theory of mind and mentalizing. So here you can see, if you look up this video, which is linked here, it shows different examples of the type of theory of mind test used in children whom the theory of mind capability emerges in the 4 years age. And just to give you a very quick example of the kind of theory of mind tasks. One involves looking at a cartoon, and there's one protagonist in the cartoon that places some item into a hiding place so that another protagonist is seeing this. And then as the other protagonist leaves the room, this first protagonist secretly, secretly from the other protagonist, changes the location of this item to some other hiding place. And then the other protagonist, who is not aware of this, enters the room. And then the question is, where will this other protagonist look for this hidden item? And children who are not old enough, they don't yet have this theory of mind capability. They will say that this protagonist will look in this second place, because they don't understand that. Okay, so this person who left the room, protagonist, doesn't have knowledge that the item had been transferred. So they think that this other protagonist has the same knowledge as they have, as the reader of this cartoon. But then children who have passed the theory of mind, age, have acquired the capability of theory of mind, they will say, well, this other protagonist doesn't know that the item has been moved, so he thinks it's in the first place, so he will look in there. And this is a kind of sign that tells the experimenter, okay, so this kid has the theory of mind capability. Also, theory of mind is needed when detecting deception by others, and deceiving others, because in order to tell lies, one has to understand that the others don't know what one knows. Okay, so these are examples of the kind of theory of mind tasks that have been used. And so when it comes to theory of mind mentalizing, there are these influential views. There's the simulation theory and theory. And these are closely related concepts to emotional empathy and cognitive empathy. So simulation theory basically means that we understand others via self-reflection. And okay, so this is a very fundamental mechanism when we think the other person is like us, and a member of in-group. And then this theory, it's an alternative mechanism where we use this knowledge of how do people in general act in different circumstances, and this general semantic knowledge about how others behave. This is used when we interpret actions by outgroup members. And okay, so this is division of the theory of mind processes into simulation theory and theory. But theory of mind, according to a recent very interesting article, should be broken down to even more fundamental parts. So as stated by these researchers in their 2015 TICS paper, the uses of their term theory of mind has exploded across fields ranging from developmental psychology to social neuroscience, psychiatry research. However, the meaning is often vague and inconsistent. The biological bases are subject of debate, and the methods used to study is highly heterogeneous. And the original definition does not permit easy downward translation of more basic processes, such as those studied by behavioral neuroscience. And because of this, the interpretation of neuroimaging results is left quite vague. And then these authors argue for a reformulation of theory of mind through a systematic two-stage approach, beginning with a deconstruction of the construct in the comprehensive set of basic component processes, followed by complementary reconstruction from which a scientifically understandable concept of theory of mind can be recovered. And these authors don't really, complete this process. They just propose some ideas of how to do this. So this is an ongoing task, but they do back up their claim by some analysis, and they present some very interesting viewpoints. So first of all, there's confusing about theory of mind, and this is due to many publications that have treated theory of mind as a monolithic process. So they're using different kinds of theory of mind tasks, and then they kind of consider that they always activate the same network or some single set of brain regions, and then they fuse together this varieties of theory of mind. And so here there's first of all this pooled meta-analysis, and you can see effect size. I mean, this identifies certain regions as candidate for theory of mind. Some of you who are perceptive can perhaps see that this is really overlapping the default mode network quite nicely. But then looking across these different task groups, different kinds of theory of mind tasks, and suddenly, for example, the posterior STS comes up as something that is involved, across a number of task groups. So okay, here the finding is not identical to the meta-analysis that I showed in a few slides ago. So there's a lot of unexplored regarding the theory of mind, and these authors also point out that after the simulation theory and theory, some psychological level distinction of theory of mind were presented. So, for example, the rapid, fairly automatic theory of mind is called the implicit theory of mind, versus slower, more deliberate, effortful evaluation of others, which is explicit theory of mind. And okay, so we can relate to this situation. Sometimes we get it very implicitly. We don't even stop to think about it, and sometimes we quite carefully consider what is going on here. Also cognitive and affective theory of mind, mentalizing about self versus other, and so on and so forth. And now they then go ahead and propose this deconstruction of theory of mind by asking the question: what are the basic constituent processes that, when combined, might underlie different types of theory of mind abilities? And so here, this is just a suggestive list. So perception and valuation of socially relevant stimuli, for example, are the faces, what's the gaze direction, interpretation of interoceptive signals, elicited by socially relevant stimuli. How do we feel? And this relates to this concept of social marker proposed by Antonio Damasio. And this is memory, access to relevant semantic or conceptual information, executive processes, motivational processes, causal inference processes. And then here you can see a figure from their publication, how to deconstruct and how to reconstruct. So going from predicting behavior based on attributed mental states, you can find these intermediate functionalities, ability to separate belief from fact, self-other distinction, and so on and so forth. and then you get to this very basic processes like view invariant representation, which we know, takes place, after the striate cortex and on a higher visual processing hierarchy levels. Then something which also is a very important concept in the area of social cognition are the mirror neurons. So the mirror neuron system was first discovered in macaque ventral premotor area F5 by Giacomo Rizzolotti and colleagues. And here I mention a YouTube video, and at 0.4 minutes and 35 seconds, if you follow this link and scroll on to that, neurons that fire while seeing an action, executive action, is very beautifully illustrated. You will hear this rattling sound, and this is the action potentials fired by a neuron, and you can really see that this monkey, when he reaches, this neuron fires. Also, when he sees another person reach, this neuron fires. It's very clear. You don't need any statistics. And then is further studies, they've noticed that some mirror neurons fire during and seeing a holding of an object but not grasping, and vice versa. Some only fire during goal-direct behaviors with an object but not when such are executed without an object. So these could be basic neural mechanisms for coding of others' intentions, for example. And some neurons fire when cracking a nut, seeing another crack a nut, and hearing another crack a nut. So I mean, these mirror neurons sort of link this motor executive system to social cognition. Also, some mirror neurons code for location of the action in space. Some code actions in view-dependent manner. In humans, inferior frontal gyrus and posterior parietal cortex are activated during observation and execution of actions. The insula is activated both during feeling of disgust and when observing others who express feelings of disgust, and the same for experience and observed pain. And so it seems that the mirror neuron system also evolved emotions, something which would be more challenging to study with animal models, because how do we know what the animal is actually feeling? Because we can't really ask the animal. And then there's a link between the mirror neuron system and theory of mind and empathy, and this could be one of these mechanisms of theory of mind that would allow deconstructing and reconstructing. And we also know that there are mirror neuron system abnormalities in individuals with autism. Here you can still see a figure from a publication showing this key and sub mirror neuron system region, and the more important the region, the larger the circle, and you can see the abbreviations of the anatomical regions as well. And okay, then to go on to a different kind of experimental example, how can we study social cognition in the laboratory? So some years ago, we set forth to question and answer is perception of social interaction shaped by knowledge of genetic kinship. And the social science question here is so-called kinship premium effect, which in behavioral studies have been identified as willingness to exert more effort on behalf of kin versus emotionally as close others. But okay, so the behavioral experiments, they've left out some open questions. And this is why we set forth in a collaborative effort to study this with neuroimaging. So I would say, in addition to asking the subject, as we do in behavioral studies, we also ask the brain. So I mean, does the brain see this differently? And so the experimental subjects, they were watching a movie called My Sister's Keeper. The other sister is sick with cancer. The other is healthy and is to donate a vital organ to the sick sister. And the sisters are seen as genetic versus adopted on different runs. So we just manipulated this tiny bit of a prior information. These sisters you're going to see are genetic sisters. And then a different run, these sisters you're going to see in this movie, one of them was adopted at a young age. So they're sisters without or with genetic relationship. So, I mean, two genes matter in this question of kinship premium. And so then we used a method called intersubject correlation that was first introduced by Uri Hassan and colleagues in 2004. We also expanded this in 2008. And okay, so there's also a relic on this slide, a referral to seminars from some previous year that I've been giving this lecture. Anyways, this method has been introduced in various sources. So if you just do a search, you can, and if you're interested, you will also find this intersubject correlation toolbox. And if you're running your own experimental studies, you will be able to download it's freeware, and then you're gonna be able to do some data analysis using this method of intersubject correlation. And so this is based on first aligning the brains of individual subjects based on anatomical MRIs, and then for each voxel, three-dimensional picture element or volume element, we will get the time course, and then we calculate the pairwise correlations across all these time points, and then take the average, and we arrived at the intersubject correlation map. It's also possible to do this in combinations, use bootstrapping and do combinations of different number of subjects, like 15 subjects, take their average, and then shuffle the subjects who are involved in these 15 subjects, and this way you will get the better signal to noise ratio. And okay, so then you can calculate the contrast between genetic and non-genetic, in our case, viewing conditions. And okay, so first of all, we had some behavioral measures. There were no differences in self-reported emotional valence and arousal. The sisters here were perceived as genetic, indicated by red color. The blue color indicates the adopted case, and as you can see, emotional valence, is it good, high ratings, is it bad, and neutral in between. And you can see these ratings go very nicely, and there's no differences between groups. Also for arousal, from very low to high, no differences between these two viewing conditions. Then we also measured their heart rate. There were no significant differences, as well as breathing rate, no significant differences between the scans. Also, we had subjective self-reports, and it seems genetic and adopted viewing conditions did not make any differences. So after the experiment, 90% of the subjects self-reported a genetic relationship was not relevant to them. We also ran implicit association tests, on which you can also find information in other sources. If you Google, how is this test done, and what does it inform us? But basically it tells, is there some kind of bias against, one or the other concept in our brains? And no, there didn't seem to be any bias against adopted sister either. But there were robust differences in brain activity patterns when viewing genetic brains from non-genetic sisters on the screen. As you can see on the lateral surfaces of the brain, intersubject correlation or intersubject similarity of how the subject's brains were working was significantly higher in the areas shown with warm colors here, red and yellow, and then with blue colors, the correlation was higher in the non-genetic viewing condition. So you can see, I mean, as is the brain is in a very different mode when they're watching these genetic versus adopted sisters on the screen. So it seems, yes, it does make a difference whether we're seeing the sisters in the movie as genetic or one of them as adopted at young age. Here you can see still the medial structures, and you can see, okay, there's precuneus, there's ventromedial prefrontal cortex very nicely. So again, default mode structures of the brain are functioning more similarly across subjects when they're seeing these genetic sisters in this, or the sisters in the movie as genetic ones. And so one way to interpret this is probably that perhaps this information that they are adopted at young age, this may lead to more idiosyncratic thoughts, interpretations, and we might not be aware of this. I don't think that our experimental subjects were lying when they said it doesn't make any difference how they on how they saw the moral dilemma that was unfolding in the movie. But still, it might change our cognition in subtle ways. How do we assess, evaluate this situation? And this finding, I think, is really exciting from the perspective that, okay, so there's added value in asking the brain in addition to asking the subjects when we have the social science questions. And we can this way start building a bridge between neuroscience and social sciences. I'm really excited about these kinds of findings. And okay, so then moving on to sort of clinical dimension. There are these so-called autism spectrum disorders, and as indicated by DSM-5, autism is a developmental disorder characterized by difficulties with social interaction and communication, and by restricted and repetitive behaviors. And there are specific problems with social cognition in this disorder. The economic relevance of autism is quite staggering. It's been estimated that autism costs the UK more than heart disease, cancer, and stroke combined. And okay, so this, of course, depends on how one calculates, but given that autism affects about 1% of the population and is a lifelong condition that requires treatment care, so maybe this is how the cost also accumulates to be as high as here in this estimate. And then just to give you an idea of what is autism. So there's this autism quotient questionnaire, which is also used in diagnostics. And there are some example statements which the person is to definitely agree, slightly disagree, or differently disagree. For example, I find myself drawn more strongly to people than to things. So if one definitely agrees, so then one doesn't appear as being autistic. But for example, when I'm reading a story, I find it difficult to work out the character's intentions. And if one definitely agrees, so I mean, this is sort of a reverse question. Then this is a sign, that the loading on autistic personality features is going up. For example, then 48, I'm a good diplomat, definitely agree. So then this again points towards the person not being terribly autistic. And okay, so we had this movie that we were showing. It was a sort of It was a movie by Match Factory Girl by Aki Kaurismäki, and or maybe it was Mika Kaurismäki. No, I'm not sure. Okay, but anyways, it was a Kaurismäki movie. It's like these brothers who made these movies, film director brothers. But one of the brothers has the Match Factory Girl, and the style in this movie is very reduced social interaction. So there was something that the autistic patients found sufficiently comfortable to watch in the fMRI scanner, and then there was higher intersubject correlation with brain activity, neurotypical than individuals with high functioning autism during viewing a drama movie with these social interactions. In these brain regions that I show with warm colors, and again, you can see there is some involvement of so-called social brain regions. So they are working more idiosyncratically in the neurotypical subjects than in the autistic subjects. And you may recall this figure from one of the early slides in this lecture showing how the autistic person pays attention to different things in the movie than the neurotypical subject. So then this finding sort of supports this in the social brain, also other brain regions were more similarly functioning in the neurotypical subjects when they were viewing these movies. And we did a further analysis, and okay, again, there's a referral to seminars from previous slides, previous time that I have given this lecture to university students in my lecture series, but okay, so this is not relevant for this YouTube video. But you can see we had neurotypical controls in high functioning autistic subjects, and then we ran graph theoretical analysis to identify this social and this functional subnetworks of the brain, and then we compared this across these different groups. And what we found was there were specific subnetworks that looked quite different. You know, there was this ventrotemporal limbic subnetwork, as we named it. It's split up in neurotypical, I mean, the high functioning autistic subjects, and this actually then correlated also with the level of autistic features. And so, as conclusions, eye movements play an important part in gathering of social information. Individuals with autistic personality differ in their eye movement patterns in that they fail to pay attention to social cues. And default mode or resting state network may be a social daydreaming network, which may serve the purpose of generating scripts of possible social interactions that are then available when the need arises. The posterior superior temporal sulcus seems to be the hub of perception of social cues that occur over shorter timescales and do not yet drive mentalizing or theory of mind. It is important to try to understand theory of mind in light of the basic constituents, and mirror neural system originally described for motor system also involves emotional empathy. The autistic brain processes social interactions more idiosyncratically than neurotypical, and complex network-based analysis of brain activity can yield further insights into brain communication in relationships. So I thank you very much for your attention, and stay tuned for some further lectures on this channel on cognitive neuroscience.